Synphane is a third-year pharmacology PhD candidate at the University of Virginia. She received her bachelor's in biology with a minor in chemistry from North Carolina Agricultural and Technical State Institute in 2020. There, she joined the Research Training Initiative for Student Enhancement Program, an NIH-funded initiative that supports historically underrepresented students in research projects and professional development. There, she studied protein signaling disease pathways and examined the extent of the reaction certain chemicals have with a protein that can change its structure. Synphane then transitioned to another NIH-funded initiative called the Maximizing Access to Research Careers Program, where she performed structural analysis on variants in a human enzyme that alters amino groups and identifying how mutations alter the structure and activity level of the enzyme. As a PhD candidate at the University of Virginia, her research interest involves the role of microglia in neurodevelopmental pathologies. Synphane, it's all yours.
Hello, everyone. Everyone can see my screen, okay? My name is Synphane Gibbs-Shelton. I am a pharmacology PhD candidate in the AO lab at the University of Virginia Brain Immunology and Glia Center. I really appreciate this opportunity to present, so thank you to the organizers that put this together. Today, I will be talking about microglia and their roles in different seizure models. Most of the data I will be showing you is from my paper that came out in Glia. This work was done in collaboration with members of the AO lab and the Perez-Reyes lab, both at University of Virginia. First, I wanted to give a brief introduction on seizures, starting with some facts and statistics of seizures. Seizures are abnormal excessive synchronous neuronal discharges in the brain. These abnormal discharges are typically started when there are excitatory neurotransmitters or less inhibitory neurotransmitters. Seizure disorders affect 65 million individuals globally. And the estimated cost of epilepsy is approximately 28 billion per year in the United States, making this a global and national health concern. And with 30% of epileptic individuals having drug-resistant epilepsy and most anti-epileptic drugs and research being focused on neuronal mechanisms of excitability, there is a rising need for investigations into alternative and/or complementary mechanisms that contribute to seizure regulation outside of neuronal mechanisms. Neurons are essential to the development of seizures. However, they do not function independent of other cells and other cellular factors. There's a network of glial cells that are traditionally considered the supporting cells of the central nervous system. They are critical for the proper neuronal development and function. In the brain, there are several microglial cell types like oligodendrocytes. Oligodendrocytes are the myelinating cells of the central nervous system and provide insulation to speed neuronal messages. And astrocytes guide nerve cell growth and regulate blood flow and can phagocytose synapses. Our lab focuses primarily on microglia. Microglia are the primary resident immune cells of the brain. Homeostatic microglia are highly ramified, using their processes to extend in and out to constantly patrol the central nervous system microenvironment to respond to pathogens and damage. When there is damage like seizures or traumatic brain injury, they become more amoeboid-like in structure and start releasing cytokines and engulf pathogens or debris. Previous studies suggest that microglia either promote seizures or dampen neuronal hyperactivity when eliminated by pharmacogenetic approaches. In this paper done by Wu and al., Wu shows that microglial depletion exacerbates seizure severity in acute and chronic seizures. In this study, they depleted microglia using deuteriotoxin and used kinetic acid, a glutamate agonist, to assess seizure severity. They then assessed seizure severity using a Racine scoring criteria over a span of 2 hours, and their results suggested that the depletion of microglia using deuteriotoxin worsens seizures and that microglia may contribute to epileptic phenotypes. However, concerns about pharmacogenetic elimination approaches like the thiotoxin have arisen with evidence that they disrupt structural brain features, brain behavioral and cellular abnormalities, as well as increased cytokine release and glial activity. These results confound the interpretation of the previous findings that microglia play a beneficial role in seizures. Therefore, there needs to be further investigation. In this current study, we used a pharmacologic approach called PLX-3397 that eliminates brain microglia without altering either cytokine levels, glial reactivity, or affecting behavior. PLX-3397 is a small molecule CSF1R inhibitor that we feed mice in a chow. Because CSF1R is important for microglial survival, the microglia rapidly deplete in the central nervous system in the span of 7 days. Here are GFP-labeled microglial cells during 0 to 7 days. And as you can see, initially being fed the child, there are a high amount of microglia because they cover the brain, but after 7 days, the remaining amount of microglia is near 0%. At these 7 days of PLX or control treatment, we measured mice weight. There was no difference in the weight of mice fed the control chow or the PLX chow, suggesting that this PLX approach may not make the mice ill and does not alter weight. Then at 7 days of PLX or control treatment, we measured mice open field behaviors. We placed the mice in an open field area where each mouse was placed adjacent to the wall of the arena and then able to freely explore the arena for 10 minutes under video monitoring. By itself, PLX did not affect distance traveled or mobile activity in an open field, suggesting that PLX-3397 alone does not alter open field behaviors or motor function. Next, mice were treated with either control or PLX chow for 7 days. Then they received kinetic acid treatments. Then their seizure severity was assessed using the Racine seizure scoring system. That was also done, and we went all 0 meaning normal activity. 1, the mice are very still, very immobile. 3 is when their paws start to drum. 4, they do a lot of rearing and falling, trying to get back up. 5 is when they stay down, just continuously seizing. And 6 is seizure spills, which are intense jumps. We let the mice seize for a span of 4 hours to get the area under the curve for both groups. And we saw that the PLX treated or microglial deficient mice showed worsened seizure severity to kinetic acid when compared to the control treated or microglial sufficient mice. Microglia regulating seizure phenotypes have been explored mainly in chemoconvulsive seizure paradigms like kinetic acid, which we just used, or pilocarpine. And it has been previously shown that our PLX microglial depletion approach using kinetic acid has been done in other studies, and now we used it in our new PLX approach. And this suggests that microglial depletion using our approach works. However, there has not been explored an alternative seizure paradigm. So a lot of other seizure paradigms have not been explored yet. And chemoconvulsive models have limitations. Like seizures don't typically happen over a span of 4 hours. And this model isn't as physiologically relevant as other models. And because microglial's roles in alternative seizure paradigms also have remained largely unexplored, we will now use our PLX approach in electrical and hypothermic seizure models. To extend our findings, To an electrical model, we collaborate with the Perez-Reyes lab to employ a continuous hippocampus stimulation, or CHS. Following implantation of EEG electrodes, we treated mice with PLX or controlled chow for 7 days, then performed CHS. Here, microglial elimination accelerated the progression of status. It accelerated the appearance of discrete seizures using CHS and also accelerated animal mortality. Furthermore, we used a recently developed VGAT-CRE mice model of temporal lobe epilepsy, where VGAT-CRE mice develop spontaneous seizures following mild electrical stimulation to induce kindling. And the PLX-treated VGAT-CRE mice experienced a longer duration of evoked seizures, which is shown in figure F, and displayed an acceleration to a fully kindled state, which is having 5 evoked convulsive seizures. Together, these results indicate that microglial elimination facilitates the development of electrically induced seizures. Finally, we sought to translate these findings to a more clinically relevant seizure paradigm. So we use a hyperthermic model. Febrile seizures or hyperthermia-induced seizures are most common in neurological disorders globally in children. They occur in about 2% to 5% of the children in the United States and Western world. Furthermore, a subset of these prolonged seizures can affect long-term effects on children, including developmental delays, making hyperthermic seizures an interesting model to look at. We developed a febrile seizure system where we expose postnatal day 13 to 15-day-old mice to increase temperatures in a hypothermia chamber to elicit behavioral convulsions at 41 degrees Celsius. We injected mice, like young pups, with PLX 3397 over a span of 3 days. So they were injected around P10 and P12. We got baseline activity from mice that was first recorded for 30 minutes at around 35 degrees Celsius, following which hypothermia was induced by subjecting them to temperatures around 41 degrees Celsius for 30 minutes or until seizures were induced. We saw that hypothermia increased CFOS expression, which was further increased by PLX-induced microglial elimination. And in the developing mice that were microglial depleted using our PLX approach, they displayed convulsions earlier on, and a greater percent of mice show conversions at 41 degrees Celsius, and greater mortality rates from hypothermic exposure, indicating exacerbated hypothermia-induced seizures and the absence of microglia using our PLX 3397 approach. Taking all together, our results from our chemoconvulsive model, electrical model, and hypothermic-induced seizure models with our microglial elimination approach comprehensively suggest beneficial roles for microglia in acute seizures. Given the evidence that microglia playing beneficial roles in primary initial seizures, we sought to investigate the possible roles for microglia in secondary or subsequent seizures. We did this by using our chemoconvulsive models, where we had 2 groups that were treated with controlled chow for 7 days and exposed to kinetic acid. One group was then maintained in controlled chow, and the second group in PLX chow to deplete their microglia. After another 7 days of their respective chow treatment, mice were treated with a second round of kinetic acid, and then their seizure scores were monitored over time. Their seizure scores were similar between microglial sufficient mice that experienced a single or 2 rounds of kinetic acid seizures, as well as between microglial deficient mice that experienced a single or 2 rounds of seizures, indicating that the initial kinetic acid-induced round of seizures does not increase the effect of a second round of kinetic acid-induced seizures. However, in the second kinetic acid-induced seizure, Mice that lacked microglia between the first and second bouts also showed a greater seizure severity compared to mice that were microglial sufficient. Furthermore, with the help of the Perez-Reyes lab, we extended these studies to a paradigm where electrically induced seizures can generate spontaneous recurrent seizures and begat Cree mice. To this end, electroimplanted mice were kindled using a hybrid kindling paradigm and then allowed to recover and develop spontaneous recurring seizures over a 2-week period while spontaneous recurring seizures were measured. The same as the findings before with the kinetic acid, PLX-treated mice showed an increase in the number of spontaneous recurring seizures in the chronic phase. These results, both in chemoconvulsive seizures and electrical seizures, indicate that microglia also play beneficial roles in subsequent and secondary seizures following an initial seizure. Next, we want to answer the question, are repopulated microglia beneficial in seizures? The reason being is that it has been previously shown that repopulated microglia can be beneficial in different neuropathologies, like eliminating and repopulating microglia following PLX-induced elimination is neurobeneficial with aging, also following intracerebral hemorrhage. Therefore, we speculated that repopulated microglia may have a stronger seizure limiting potential than endogenous microglia. To this end, we treated mice with either control or PLX chow for a week and then took away the chow so that the mice were able to repopulate their microglia. And here is the image before of day 0 when they are lacking their microglia after 7 days of PLX treatment, and then their microglia is continuously growing back to be about the same as it originally was. After eliminating and repopulating the microglia, we can then expose all mice to kinetic acid. Seizure severity was similar in mice with endogenous and repopulated microglia. And because microglia are known to progressively mature during repopulation following PLX treatment, we then asked the question whether mature repopulated microglia could show a more protective effect by conducting kinetic acid-induced seizures 4 weeks after PLX treatment. Then microglial maturation following PLX is complete. As with 1 week, mice with repopulated microglia at 1 month similarly seize like their counterparts at 1 week of repopulation. In summary, our findings are consistent with the recent observations that microglia dampen seizure and network excitability, and more importantly, our PLX method that reduces the minimal of the effects of microglial depletion also shows these findings. Microglia play roles in different seizure paradigms like chemoconvulsive seizures, electrical and hyperthermic seizures, and microglial play roles in subsequent and secondary seizures following an initial seizure. Finally, endogenous and repopulated microglia play seizure limiting roles in chemoconvulsive seizures. And with that, I would like to thank all the authors involved in this paper, my committee, and special recognition to Dr. Sharma. And now I will open the floor for questions.
Thank you, Synphane. That was a great talk. If anybody has any questions, feel free to raise your hand or put them in this chat. But I think I'll start off with one. Synphane, we think of these microglia as, like as you said, as the phagocytic cells that clear up debris and everything from the brain. And in epilepsy, I'm thinking of them, I don't really know the beneficial role that they would be playing. And I was just hoping you could speculate on the thing that I'm kind of thinking about is, are these microglia targeting these cells that are focused in seizure onset to help reduce severity of the seizures? Do you think they're targeting specific neurons, or are they targeting some other debris or anything else that's floating out there. If you could just speculate on that.
That is a great and reoccurring question that I always get because no one knows the mechanism of it yet. But there has been a lot of studies, or a lot compared to the overall field, showing that microglia in its actual action of processing, extending its processes throughout the brain and surveying the brain, like the amount of, the more that it surveys the brain, the more neuronal excitability gets dampened. So my theory is maybe it's interacting with these neurons in some way and somehow making sure they're calmed down or not being too excitable, because when microglial surveillance stops, there is an increase of neuronal hyperexcitability.
Gotcha. Thank you so much. Trevor asked in the chat, do microglia protect via interaction with astrocytes?
That is a great question as well. I believe that microglia is not the sole glial cell helping to interact with neurons and reduce seizure severity. So I do believe there could be a community effect of glial cells working together to protect neurons.
There are no other questions. I think it's about time to wrap it up. Synphane, thank you again for your talk, and Christos, thank you for yours.